So if at all anybody tries to join, we'll just let them in during the course of the meeting. Right, for now, I'll just share my screen so that we can actually begin with the session. Yeah. Okay. So, hello everybody. Welcome back to this ongoing course that is basics of neurobiology, basically. Right. So over here we have been going through like what is neurobiology, how neurobiology and neuroscience are a little different from one another, how over time this particular subject evolved, right? And what was the kind of techniques that were developed over time to actually study the subject. So these are the few things that we have already gone through. Now we will begin with the actual part of neurobiology, which begins with a little bit of physiology, right? So what we'll be discussing in this particular session has actually not been picked particularly from these two books. A little bit of it has been extracted from there, but most of it has been picked by from actually a physiology book, which is Tortora, right? That is a book for animal physiology, right? So over here we will be looking at the components of the nervous system, how there are two major parts to the nervous system. One is the CNS, the other is the PNS. How there are variations between these two parts, because further in our course, we will focus mainly on the central nervous system. But sometimes, because most of our sensory receptors, most of the world that we are perceiving, etc., is responding to the rest of our body. So PNS is actually also playing a very vital role, right? So we need to have a basic knowledge about what is PNS, where it is, right, and how it is different from the CNS, central nervous system. Also, we'll discuss about parts or the cells that are there within both the central nervous system and the peripheral nervous system, and again, how they are different from one another, and in what ways are they similar also, right? So that is what we are going to discuss in this particular session. So, we can begin with the components of the nervous system. This is something we were supposed to pick up in the last session. So, we had left off over here because our discussion for that history expanded a little bit, right? Because we also discussed some techniques, some experiments in between. This is where we will begin today. So when we talk about components of the nervous system, it we'll come to it. But what is the nervous system, first of all, right? So it is one out of 11 systems that are present in our body, right? And it is actually one of the smallest systems, but it is most complex, right? So the largest system in our body is skin, right? It is not the smallest system, but it is one of the smaller systems, right? It's like by mass, if we see, it is only 2 kg of our entire body, right? And it is about 3% of the total body weight, right? So, the nervous system is again one of the smallest systems. Apart from this, another very, this is something I learned in PhD when I had started doing or started, looking at the nervous system actually, and started looking at the brain. That you might have heard this, that the source of energy for brain is primarily glucose, right? So our body can still use alternative sources of energy, but our brain can only use glucose, right? So, for instance, keto diets are said to be bad in the long term for the brain, right? Because those ketone bodies will starve your brain also, right? So, in relation to that, I was reading once when I was preparing for my comprehensive exam over here, that brain, it is although it is only 2% of the body weight, over here it's written 3%. So 2 to 3% of the body weight, still it demands 20% of the entire glucose, that is there in the body, right? So 20% of that entire glucose is actually goes to the brain. So it is functioning so intently and so intensely that such a huge portion of the body glucose, the blood glucose is actually dedicated to the brain itself, right? Also, this intricate network of billions of neurons and even more neuroglia is organized into two main subdivisions, right? So, what are these subdivisions? The central nervous system and the peripheral nervous system, right? So, these are broader subdivisions that we are aware of, right? So central nervous system, we have studied this in school also, right? So that central nervous system comprises basically of the brain and the spinal cord. Peripheral nervous system comprises of the nerves that are there, right? The certain sensory neurons, etc. Right? So, yeah, so this is the CNS, that is the brain and the spinal cord, and this is the PNS, which is cranial nerves. Spinal nerves, ganglia, enteric plexus in small intestine, and sensory receptors in the skin. Now, this is one of the figures that I have picked from Candle itself, but I would also like to point out that not all cranial nerves are actually a part of the PNS, right? There are some cranial nerves, I think the optic and the olfactory nerves, 1 and 2 or 1 and 3, I think, which are actually a part of the CNS, right? Because they are actually directly connected to the brain. They don't go through any other route, right? So that is why some cranial nerves, two, I think, olfactory and optic nerves, are actually a part of the CNS, right? So, the central nervous system consists of the brain and the spinal cord. The brain is a part of CNS that is located in the skull, right? So, this is the skull, the bony structure of our head, and this is where CNS or the brain is basically located, right? And it contains about 85 billion neurons, right? So, that is actually a huge, huge number. These are the number of neurons that roughly is present in all of our brains, right? So the spinal cord is connected to to the brain through the foramen magnum of the occipital bone. So occipital bone is somewhere over here, right? And there is some region over there which is known as foramen magnum, right? Which is basically connecting the spinal cord to the brain, right? And it is encircled by the bones of the vertebral column, right? So this foramen magnum, if I'm not wrong, this all of a sudden, like while preparing notes, also it did not click my mind. But right now, when I read it again, it did strike my mind that this is actually, I think, the bone that breaks if you are hanged, right, in a death sentence or something. If you are hanged, this is the bone that breaks and enters the cerebellum or damages the cerebellum, due to which a person actually dies, right? If I'm not wrong, this is the one. So the spinal cord, it basically contains about 100 million neurons, right? So again, a huge, huge number over here again. Uh, so the central nervous system processes many different kinds of incoming sensory information, right? So what is this kind of information? It could be sensory information related to touch. So you have touched something hot, you are smelling something, you are looking at something, you are trying to visualize it. All these are kinds of sensory information, right? You are listening to some music, right? So it is also the source of thoughts, emotions, and memories. So most signals that stimulate muscles to contract and the glands to secrete originate in the CNS, right? So CNS is actually controlling the entire body, right? It through, like, even if it is PNS, it is some part of it is supported by central nervous system, right? Later we will study an autonomic nervous system also. So some part of that is also controlled directly through the CNS also, right? So CNS is the kind of like the headquarter or the head office, right? It knows what everything else is doing, right? It doesn't have to be the primary respondent sometimes, right? For instance, if there was a head office for a particular brand in New Delhi, right? And there were multiple other offices, one of which was maybe in Nagpur, one was in Bombay, one was in, Kolkata, one was in Dehradun somewhere, right? And if there was a problem that arose in Dehradun, that particular office will respond, right? But if the problem escalated, it will reach the head office also, or the head office will have a knowledge about all the respondents, but it doesn't have to be the primary respondent, right? Because if you wait for a problem to be resolved through the head office, it will be a longer procedure, right? So this query would have to go to the that particular local office. Local office will escalate it to the head office. Then head office will think about it. They will go through their own procedures. Then they will revert back to the local office. Local office will again revert back to the person who raised the query. So it is a longer process, right? Sometimes the need of the hour is not a longer process, but a quick resolution, right? So in that case, this PNS, ANS themselves can also manage. But CNS comes into picture whenever there is something crucial that is involved, some thinking, some cognition that is involved, right? So the peripheral nervous system consists of all the nervous tissue outside the CNS, right? So any nervous tissue that is present outside of the CNS or central nervous system is a part of the peripheral nervous system, right? It contains nerves, ganglia, enteric plexuses, and sensory receptors, right? So all these are components of the nervous system themselves, right? So first, let us understand what the components of the PNS, right? Brain is anything way something that we are going to talk about at length, right? Similar is the case for uh, not spinal cord. I think we won't talk about that much, but brain we are going to discuss at length eventually, right? So first, let us get through with our knowledge about PNS, right? So that we can move ahead, right, to what is the main focus of this particular course. So when we talk about PNS, as we read, there are multiple parts of PNS, right? Some of them are nerves, some of them are ganglion, etc. Right? So when we talk about nerves, what is nerve? It is basically a bundle of hundreds of thousands of axons plus associated connective tissue and blood vessels that lie outside the brain and the spinal cord. Right? So that is the definition of what a nerve is. Right? Uh, just to give a disclaimer, we have a tech fest going on at our institute right now. So if at all there are background noises, I am really apologetic for that. On Sunday, probably I'll have the class at a different location so that these noises don't bother us. Today I had not thought it through. And this fest is also, burdening, adding up to that sound. Yeah. So that is what a nerve is, right? So it is not just a collection of axons, but over here it also says that along with the axons, it also is a collection of the connective tissue that is present around it and the blood vessels that are supplying these axons or these neurons, right? So all this together forms the nerve, and nerve, it is known as a nerve when it is present outside of the brain and the spinal cord, right? So, an important thing to know over here is if it is present within the brain and the spinal cord, right? This complete this complete network of the axon, of the connective tissue and the blood vessel together, then what will it be called over there? It will be called a tract, right? It will not be a nerve anymore. It will be known as a tract. So in the PNS, it is known as a nerve. In the CNS, it is known as a tract, right? TRACT, tract. Yeah. So there are 12 pairs of cranial nerves which emerge from the brain and 31 pairs of spinal nerves which emerge from the spinal cord, right? So the cranial nerves go from the brain to the eyes, ear, mouth, other parts of the head. Central nerves are in the brain and the spinal cord themselves. Peripheral nerves go from spinal cord to arms, hands, legs, feet. And autonomic nerves go from spinal cord to, the internal body organs such as lung, heart, stomach, intestine, bladder, sex organs, etc. Right? So each nerve basically follows a defined path and serves a specific region of the body, right? So if we are, I think vagus nerve is one of the cranial nerves which spans a lot of different body organs, right? Some part of the face, some part of the respiratory tract also. Uh, and some other, some facial muscle also, right? So different parts it spans. Otherwise, generally, these nerves serve specific regions, right? They are specific to a region. Then we can talk about ganglia, right? So, what is a ganglia? These are small masses of nervous tissue consisting primarily of the neuron cell bodies, right? So, the axon part of the neuron is going and forming a nerve or a tract, right? In the PNS, it is forming a nerve. Over here, the cell body part of the neuron, right? What is that forming? It is coming together to form a ganglia. So it consists primarily of the cell bodies that are located outside the brain and the spinal cord, right? Again, if it is within the CNS, within the brain and the spinal cord, it is not known as a ganglia, it is known as a nuclei, right? So ganglia are closely associated with cranial and spinal nerves, right? Then there are enteric plexuses, which are also a part of the PNS. So enteric is a word that has something to do with the digestive system, right? So enteric plexuses basically stands for the nerves that are basically going and spanning the digestive system in some way. So enteric plexuses are extensive network of neurons located in the walls of organs of the gastrointestinal tract, right? So that is what it is doing. Over here, these submucosal plexus, the mindrect plexus, all these are a part of the enteric plexuses, which again are the neurons that are spanning the gastrointestinal tract, right? And they are also a part of the peripheral nervous system naturally because they are not a part of the CNS, and everything out of the CNS, which is neuron related, is actually peripheral nervous system, right? So it is simple in that sense. So the neuron of these plexuses help regulate the digestive system, right? So, what is the function of these neurons? Obviously, because they are present in the gastrointestinal tract, they are helping in regulating the digestive system, right? Then sensory receptors, right? What are sensory receptors? They are like the nerve endings that are present on our, fingertips, on our toes, on the surface of our arm, or you know, like throughout all the sensory receptors, they basically refer to structure of the nervous system that monitor changes in the external or internal environment, right? So these all examples that I was giving were external sensory receptors, right? There are also there is also something known as proprioception, which is a little different, but also sensory receptors within our body, right? For instance, tongue is a kind of receptor, right? Although yeah, so it is a kind of receptor that is within our body, right? Or, even like if I feel pukish, that is because some kind of regurgitation feeling I'm having, some kind of anti-peristaltic movement is there. So, some part of the gastrointestinal tract has some sensory receptor that is actually, viewing that and responding to it. So, that is why I have this feeling that I will puke, right? So those are examples of the internal and external receptors, sensory receptors that are present. So examples of sensory receptors include touch receptors in the skin, photoreceptors in the eye, another internal receptor, right? Olfactory receptors in the nose, the your taste buds on your tongue, etc. Right? So this is what is skin is. It is like a representation of the skin, right? Over here, the interesting thing to notice that look at the kind of different kinds of sensory receptors that are present only in the skin also, right? So there are some kind of receptors with free nerve endings that basically sense pain, heat, and cold. Then there is something known as Merkel discs, which basically sense touch. Then there are something again known as Krauss end bulbs. These again are responsible for touch reception, right? Or touch sensation. Then root hair plexus again responsible for touch reception. Meissner corpuscles also responsible for touch sensation. Then Passician corpuscles, which are responsible for pressure sensation. So, for instance, if I put this, if I pinch my hand, it is applying some kind of pressure, right? So that kind of thing is sensed by this Passician corpuscles, and then the Ruffini endings, which also sense pressure. Right, so even within the skin, can you see how many different kinds of sensory receptors are actually present, right? And some of them have redundant functions also, right? Because they might be responding to different types of touches or different, intensity of touches, something like that. Okay. So, yeah, in summary, if we look at it, the central nervous system is consistent of the brain and the spinal cord, right? And then there is the peripheral nervous system, which is all of the nervous tissue outside of the CNS, right? Inside this PNS, it is further subdivided into three kinds. One is the somatic nervous system, one is the autonomic nervous system, and the other is the enteric nervous system, right? Uh, Earlier, even the enteric nervous system was a part of the autonomic nervous system, and there were just two classifications, somatic nervous system and autonomic nervous system, right? But now we have three subclassifications, and enteric nervous system is actually different. So when we talk about somatic nervous system, what is it? So it has something to do with, motion. So somatic and special sensory receptors and somatic sensory neurons basically connect this somatic nervous system to the central system, and motor neurons that are present receive signal from the central nervous system are basically responding over here. So the somatic nervous system, because it has something to do with movement, it is basically present in the skeletal muscle, the voluntary muscle, right? So that is why somatic motor neurons are involved, because these are neurons that fire when a decision has been made, right? Or a command is being given, right? So the brain has thought that, okay, I want to move my hand, right? So it will move the hand from point A to point B. If the brain has thought, I want to pick up that water bottle, it will move my hand and pick up that water bottle, right? If I want to just wash the water bottle, I'll just wash it, I'll keep it. If I want to drink water from it, I'll just drink water, keep it back, right? So all these are voluntary actions, right? I am well aware of what I am doing, and it is my decision to wash the bottle, it is my decision to drink water, right? So all that is a part of the somatic nervous system, right? So this is actually different from the autonomic nervous system, right? Because over here the movements are involuntary, right? So autonomic nervous system basically controls all the internal body organs, the glands, the smooth muscle, the cardiac muscle, the diaphragm, it is another smooth muscle only, right? So all the involuntary movements are being controlled by the autonomic nervous system, right? For instance, I cannot think that, okay, I want my heart to beat at 72 beats per minute, right? if I think like that, my heart will beat at 72 beats per minute. Now, that is not possible, right? That is something that is not in my control. It is involuntary, right? So, that is why it is a part of this autonomic nervous system. Another part of the brain is actually controlling the beating of the heart, and beating of the heart itself also is, it has some feedback mechanism due to which it might beat faster, due to which it might beat slower, etc. So I don't have any voluntary control over it, right? On the contrary, that might control the other thoughts that I have, right? So it is an autonomic nervous system, meaning it can kind, it has a brain kind of its own, right? uh, it doesn't actually have a brain of its own, but it kind of functions autonomously from the central nervous system, right? Because it is also involuntary. Although cerebellum in the central nervous system is also controlling these movements, it is controlling the diaphragm, it is controlling the lungs, it is controlling the heartbeat pattern. But over there, whatever muscles, whatever nerves, etc., are there, they are not under my direct control. So that is why it is autonomic, right? Then comes the enteric nervous system. So, as the name suggests, it basically controls the smooth muscles, glands, and endocrine cells of the gastrointestinal tract, right? So, enteric, something to do with the digestion, digestive organs, right? So, as I said, because this is also an involuntary motion, uh, um, you know, earlier it used to be a part of the autonomic nervous system. Now, since this itself is such a huge, system, and you might have heard of this upcoming, field of point of view, that is the gut brain axis, right? A lot of research is being going on in this direction, right? That can good microbiota, can it improve certain symptoms of even something as bad as autism, or dementia, which dementia which has caused physical damage in the brain, can even that be the progression can be slowed just by eating right, something like that. So all these things are being they are kind of talk of the town right now in some uh, neuroscience groups, right? So it is a very hot topic right now. Due to which also, and it is like this enteric nervous system actually has a huge deal of neurons that are there, that are only dedicated to the GI tract, which is why it was separated as another kind of subclassification itself, right? So then we can look at the functions of the nervous system, right? So what are the functions of the nervous system? It basically carries out a complex array of tasks. It allows us to sense various smells, to produce speech and remember past events. In addition, it provides signal that controls body movements and regulates the operation of internal organs, right? So all that it does. Apart from this, the diverse activities that it does can be grouped into three basic functions, right? It, these functions are either sensory, that is, some information is being input or put into the system. Integrative, that is, whatever information we have or whatever information has been fed to us is being thought about, is being processed, right? So that is integrative. And motor, which is the output of whatever has been asked, or output of whatever we have thought, right? So those are the diverse functions of the nervous system, right? Okay. So the sensory receptors, they detect internal stimuli such as increase in blood pressure or external stimuli, for example, a raindrop that lands on your arm, right? This sensory information can then is then carried to the brain and spinal cord through the cranial as well as the spinal nerves. Okay, so again, this kind of sensory reception that is said in the first example, that is increase in blood pressure, right, or increase in body temperature, or some such internal sense. It comes under a term that is known as proprioception, right? So that is uh, to basically have a sense of within, functions within our body. The nervous system also possesses sensory information by analyzing it and making decisions for appropriate responses, and activity which is basically known as integration, right? Once sensory information is integrated, nervous system may elicit an appropriate motor response by activating effectors which is organs that will respond to it, right, through cranial and spinal nerves. Stimulation of effectors causes muscles to contract and glands to secrete, right? So, for instance, the information that I got was uh, that my brain got was that I was eating something, and my brain received this information that okay, food has been consumed, right? So that is why it will go through, my mm, food duct. It will go into my stomach from my esophagus food pipe. It will go into the stomach. Gastric juices will be released, right? It is not a direct response of the stomach receiving the food, but an intervention of even the neural cells that are present in that enteric mindteric plexus and the other enteric plexuses, right? So as a response of those, these gastric juices will be released, right? Then my brain has this knowledge that okay, now the body has food, it is churning the food. After this amount of time, we have to stimulate the pancreas to actually release the pancreatic juice, or whatever hormone is required to for it to stimulate it. All that will be controlled basically in some sense by the nervous system itself, right? Then we'll talk about the cells of the nervous system, right? How there are two major kinds of cells that are present in the nervous system. They are the neurons and the neuroglia, which are also known as the glial cells. So neurons and the glial cells, right? So we are going to discuss about both these different kinds of cell types inside the nervous system, and we will also see how there are subdivisions in these cell types, right? So before that, please give me a moment. I need to plug in my charger and I'll fetch water. I hope this is more predictable again. Yeah. So as we were saying, there are two kinds of broader classifications of the types of cells that are present in the nervous system, right? They could either be neurons or glial cells, right? So over here, this representation has both of these examples, but the neurons over here are not marked. So these cells with these yellow colored cell bodies are neurons. Basically, they have a long axon, dendrites, everything that we are going to study about very soon. Along with that, there are other cells that are marked over here, microglia, astrocyte, ependymal cell, and oligodendrocytes, which are all basically glial cells of the central nervous. Okay, I accidentally muted myself. What I said was that all these cells that are marked are basically the glial cells of the central nervous system, right? So I hope that was not muted earlier also. Yeah. So the nervous tissue basically comprises of two types of cells. One are the neurons, the other are the glia, right? So these cells combine in a variety of ways in different regions of the nervous system, right? So there are some places wherein maybe there will be excess of astrocytes along with neurons. There will be other places wherein there will be excess of oligodendrocytes with neurons, or all three of these are present abundantly, or there will be places where only astrocytes and the neurons are present. Some microglia, some are present in the background, no oligodendrocytes whatsoever, because there's no myelination in these axons, right? So you'll learn about that. So when we talk about these cells, in addition to forming the complex processing networks within the brain and spinal cord, these neurons also connect all regions of the body to the brain and the spinal cord, right? So that is one additional function. Yeah. As a result of their specialization, most neurons have lost the ability to undergo mitotic division. So we know that the neurons are terminal cells, right? They cannot divide. Actually, all these specialized cells, any cell that has a specific function, are generally non-mitotic, right? So, for instance, our RBCs, our adult or mature RBCs, they have also lost a nucleus. They will also eventually go and die, right? They will not undergo mitosis again, right? Similarly, our Kupffer cells, which are resident macrophages of the liver, right? They're specialized, right? If they die, new Kupffer cells might be born, but they will not undergo any mitotic divisions, right? Themselves. So similarly, neurons are also specialized cells which have lost the ability to undergo mitotic divisions. Now, along with that, whether new neurons will be formed or new Kupffer cells will be formed or new skin cells will be formed will depend on whether there is a pool of stem cells that is present, right? So there are multiple hematopoietic stem cells are still there. So even if RBCs are constantly dying, new RBCs are continually formed, right? Similarly, the macrophages, they are also in that homology only, the hematopoietic ones. So they will also be formed, right? The skin cells have stem cell pool, so they will also continually form, right? Nervous system cells, such as neurons specifically, do not really have a stem cell pool of neurons, right? So that is why they do not, if a neuron dies, new neurons are not formed, right? That is the case in most of the organs of in most of the parts of the brain, right? If at all there is a stem cell pool, it is very less and it is restricted to some areas of the brain only, right? Most areas, if the neurons will die, there will be no neurons that will be formed. And because they are specialized, they have lost the ability to undergo mitosis themselves, right? Then we talk about neuroglia. They are smaller cells, but they greatly outnumber the neurons, perhaps by as much as 5 times. Right? So neurons are bigger cells, as we could have seen in that earlier representation also. Glia are comparatively smaller, but they are much more in number, right? They are almost 25 times in number than the neuron. So this big cell is a neuron, and these smaller cells, as compared to the neuron, they are smaller. Astrocyte is still big. Microglia is smaller. Ependymal cells are smaller. These oligodendrocytes are also almost as big as astrocytes, right? So these are basically uh, outnumbering the neurons and by a lot, right? What do these neuroglia do? They support, nourish, and protect the neurons, and they maintain the interstitial fluid that bathes them, right? So that is their function. Also, unlike neurons, these neuroglia continue to divide throughout the individual's lifetime, right? So these are cells that can still divide. They have the capability to divide, right? Parts of a neuron. So again, like I said, if something was specialized, it will lose its mitotic ability, right? But there are processes that can bypass that ability, right? That is not a part of neurobiology, so I will not go in detail, but there are some processes such as de-differentiation. So differentiation will create a specialized cell. De-differentiation will take that specialized cell back to an undifferentiated form or a de-differentiated form, so that it can choose its fate again. It can divide maybe and then choose its fate again. Something like that can happen, right? So when we talk about a neuron per se, there are three parts of a neuron that are present. What are these three parts? One is the cell body, the other is dendrite, and the third is the axon, right? So these are the three parts of a neuron that are there, and multiple other cell organelles, multiple other, associated structures to these three major parts. So when we talk about cell body, for instance, this first yellow colored portion, right? This bigger portion in the middle, not these branches, not these branches, this bigger portion in the middle, which has a nucleus over here. This is basically the cell body or the soma, right? So the cell body of the neuron is also known as a perikaryon or a soma. It basically contains a nucleus which is surrounded by cytoplasm that includes typical cellular organelles such as lysosomes, mitochondria, and Golgi complex. Right? So these are normal cell organelles that are also present over here. Then, when we look at parts of a neuron, the neuronal cell bodies also contain free ribosomes and prominent clusters of rough endoplasmic reticulum, which are termed as Nissl bodies, right? So, it has like RER is there in other cells also, right? And RER will actually contain, it is rough because of the presence of ribosomes, right? So what they are saying is that in neurons, along with the free ribosomes, there are also prominent clusters of RER that are present, and because it is clustered over here, it has a little different kind of shape. Right, or it is a characteristic feature actually of the neuron in that sense, right? So this is known as a Nissl body, right? Now, why this is important over here, or why this is being told at all over here is because when we look for neurons, right, in a postmortem brain sample or in a mouse brain that you are staining, or in a in one of these cases actually, right, an ex vivo animal model brain staining, or even in case of autopsy brain tissue processing. When we have to identify neurons, one of the simpler biochemical studies, like one was the silver staining that Golgi had, formulated. So silver nitrate staining is done to basically look at neurons. The other kind of staining to look at neurons that is done is Nissl body staining, right? So Nissl staining. So the ribosomes are the sites of the protein synthesis. Right. Okay, so this is something again that we already know. This is the function of the ribosome by itself. It is nothing that is peculiar of a ribosome that is present in the neuron, right? It is the general function of the ribosome itself. Also, newly synthesized proteins produced by Nissl bodies are used to replace cellular components as material for growth of neurons and to regenerate damaged axons in the peripheral nervous system, right? So two things it does. One, that whatever material it is producing, because it has rough area, right? So it has a lot of protein synthesis machinery, right? Because lots of ribosomes are there in this Nissl bodies also, right? So whatever is being produced is used to replace cellular components, right? And are also used as materials for the growth of these neurons, right? Also in the PNS, we will learn later that in CNS, regeneration of axon, etc., is very difficult. It is a very cumbersome process and it is almost next to impossible, or it is very rarely done, right? Only in some rare cases does it happen. But in PNS, however, even if the axon is damaged, often it will regenerate, right? There will be multiple cells involved in that regeneration. We will look at, I'm sorry. We will look at those cells also. And the part of the neuron that will help in the regeneration will actually be these Nissl bodies because they will have that extra protein produced only for that purpose, right? Then a nerve fiber is a general term for any neuronal process or extension. So this extension, anything will be known as a nerve fiber, right, which emerges from the cell body of a neuron. So anything is a neuronal process or an extension. Most neurons have two kinds of processes. Multiple dendrites, which are seen over here. See, these are labeled as dendrites, right? So multiple dendrites and a single axon. So from the cell body, there are two kinds of extensions that you can see at least in this figure, right? This is the cell body. From this multiple of these you can see which are known as dendrites, and a single of this thick long one you can see which is known as axon. So we can look at dendrites. One moment. Yes. Mhm. Yeah. So these two kinds of neuronal processes we were looking at, right, or neuronal extensions. One was the dendrites and the other was the axons, right? So first let us discuss about this dendrites. So dendrites are the receiving or input portions of the neuron. What does it mean? That whatever information will be given to the neuron will be through these structures or these extensions, which are known as dendrites, right? The plasma membrane of dendrites and the cell body contain numerous receptor sites for binding chemical messengers from other cells, right? So again, it does have a lot of receptor sites. Once we learn about synapse, we will understand why those kinds of receptors are important, right? Okay. So these dendrites are usually short, they are tapering, highly branched. Tapering as in they are pointed towards the end, they are highly branched structures. In many neurons, the dendrites form a tree-shaped array of processes extending from cell body, right? So it did look like a tree, right? In that earlier the structure that we were saying, they do form a tree-like process, right? Or a root-like process. Their cytoplasm does contain nestled bodies, mitochondria, and other organelles. So, because it is directly extended from the cell body itself, right? It also contains nestled bodies. It also contains mitochondria. It also contains other cell organelles. Uh, then comes the axon, which was the other neuronal extension, right? So, an axon in the single axon of neuron propagates nerve impulses towards another neuron or muscle fiber or a gland cell, right? So, what it does, what we said was dendrite is the receiving end of the for the neuron, right? So, whatever information is being transported will be received by the dendrite of a neuron. Axon is the propagating end of the neuron. So once this information is there with the neuron, it wants to, tell it to the other neurons also, or you carry it forward, right? So for it to carry it forward, there is another extension which is axon that is present, which will propagate this information to the next partner or the next neuron, right? So an axon is long, it is thin cylindrical projection that often joins to the cell body at a cone-shaped elevation called axon hillock, right? So I think I've kept a representation in the next slide, which I will point out what an axon hillock is, right? So the part of the axon closest to the axon hillock is the initial segment, right? So that is where the axon is beginning. So that is the initial segment of the axon. In most neurons, the nerve impulses arrive at the junction of axon hillock and the initial segment, and this area is called the trigger zone, from which they travel along the axon to their destination. So right now we have actually not discussed action potential or nerve impulses or how they are generated, right? So if whenever we do discuss them, I will tell you why they are basically generated at this particular part of the neuron, right? The axon hillock. So this is the part where the action potential first is generated and then it is carried forward or propagated, right? So, yeah, this is the part that we were saying. So there is a cone-shaped part, cylindrical part that is present, which joins the cell body to the this extension. It is known as axon hillock, and this is where the action potential is generated, right? This long cylindrical tube is known as axon. The axon also contains mitochondria, microtubules, and neurofibrils. Right? Oh, please forgive me. I have been having really long experiments. Okay. So, why does it contain mitochondria? It actually definitely contains mitochondria. Why? Because it has to propagate something, transmit something, right? There will be ATP that is required for that, right? For this purpose alone, it does contain mitochondria. Also, it contains microtubules and neurofibrils because there is some physical, cargo also that is transported across this particular neuron, right? This physical cargo is transported through these neurofibrils and microtubules and the motors that are present on them. right? Because rough endoplasmic reticulum is not present, protein synthesis does not occur in the hillock, right? So Nissl bodies and all are not present over here, right? They are just present over here. So this part does not have any RER, so it does not have any protein synthesis, right? The mitochondria that are present is only responsible for propagation, right? Helping in producing the ATP that will help in propagating the signal. That is its only function. So, an axon contains mitochondria, microtubules, and neurofibrils. Now, that is a repeated statement, right? Because the RER is not present, it does not occur. And okay, that is also a repeated statement actually. And the cytoplasm of the axon is called the axoplasm, right? So, for instance, the plasma that is present within the cell body is, or the cell is known as cytoplasm. The part of cytoplasm that is present in the axon only is known as the axoplasm. It is surrounded by plasma membrane, which is known as axolemma, right? So a special kind of plasma membrane is also present over here. It is known as axolemma, right? Axoplasm is contained within axolemma, right? Now, this plasma membrane may be special because it has some specific, yeah, because it has some specific additional kind of receptors or channels, which might make this axon different, right? Different from the rest of the neuron. So along the length of the axon, side branches called axon collaterals may branch off typically at right angle to the axon. So there might be branching that may take place in axon also, right? Although it is not very common, but it might take place. These are known as axon collaterals, right? Like this. It is usually at right angle from the axon. So these are the axon that is going, right? This is the end of the axon. This was where the axon began. This is where the axon is ending. And in between, there might be some collaterals that might be present like this. Then the axon and its collaterals end by dividing into many fine processes, right? Over here is the end, and you can see that it is divided into multiple finer processes, which are known as axon terminals or axon telodendria, right? So that is the name of these finer processes, which is present at the ending of the axon. It is known as either the axon terminals or the telodendria. And this is what the neuron looks like, right? So if we look at the parts of the neurons, right, there is a nucleus which is present in the cell body. The cell body has extensions. One of the extensions is known as dendrites, which are seen over here. They are tree-like, they are tapering, they are short, and they are very highly branched, right? Also, this part, the cell body by itself, and the dendrites, they contain microtubules, they contain mitochondria, they contain cytoplasm, they contain Nissl bodies, uh, etc. Right? What are Nissl bodies? They are basically rough RER granules or clusters, right? So it is an area of active protein synthesis. Highly active protein synthesis, right? Then this cell body also has another kind of extension which is known as axon. This is the axon, right? There are some parts over here that we have not discussed as yet, right? This sheath that is covering it, right? This pink colored sheath, which is known as basically the myelin sheath. We have not discussed it as yet, but we know that this axon that is going, it has a axon hillock, right? This hillock is the place wherein the cell body is basically coming, forming a cone-like structure, and it is finally forming the axon, right? We have also seen that this axon sometimes has, branching from the main body of the axon. There is some branching at right angles, which are known as axon collaterals, right? Also, these axons at the end have finer endings, right? These finer endings are known as telodendria or axonal terminals, right? And soon we will look at all these other things also that we have not already studied, which are Schwann cells. What are they? Myelin sheath, right? Node of Ranvier. So node of Ranvier is basically the place wherein this sheath is not covering the axon, right? So there are spaces in between this axon wherein there is no coverage of this sheath, and there are places where the sheath is covering, right? Now there is a logic behind that also. So if we study electric impulses or generation of action potential and conduction of action potential, we will understand why this sheath is present and why these spaces in between nodes of Ranviers are also present, right? Then again, the axon basically contains the exoplasma, which is the cytoplasm that is present within the axon, and it also contains a plasma membrane, which is known as axolemma, right? Along with this part of the neuron, that is axon, does have mitochondria, it does have neurofibrils, and it does have microtubules, but it lacks what? It lacks rough RER or Nissl body, right? So that is it for the revision of this part about what are the different parts of the neuron, right? And where they are present, right? Now, one part that we have missed is the synapse, right? Because we were talking about a single neuron. Synapse is always will come into picture when we are talking about at least two neurons, right? So synapse is basically the site of communication between two neurons or between a neuron and an effector cell. It is known as a synapse, right? So either it is two neurons, right, or neuron and effector cell, meaning there is a synapse that is present between our uh, neuron and our heart also, or a neuron and our muscle in the hand also. So that will be a neuromuscular junction, right? Uh, s- the effector cell over there will be muscle. If it is a neuron synapse, the effector cell is also a neuron, right? So that is what. So synapse is basically the site of communication between two kinds of cells, one of which at least is a neuron, right? Which is trying to tell the effector cell or the cell that is going to perform some function to do that function, right? So in this diagram, if you see, this is an example of a synapse, right? This is an axon terminal from the presynaptic neuron. This is a synaptic cleft, which is the space that is present in the synapse, right? And this is the dendrite of the other neuron, which is a post synaptic neuron, right? And you can see that there are some synaptic vessels that are present over here, which vesicles that are present over here, which will come. They will basically integrate with the plasma membrane, release whatever is there within that synaptic vesicle. Over here, it is neurotransmitter. And the receptors on the postsynaptic neuron are basically going to uh, uh, be able to bind to this neurotransmitter so that they can respond to it, right? So that is what. Both the synaptic end bulbs and varicosities contain many tiny. Okay, I think there is a slide that is missing. Okay. So there is a slide that is missing that was basically introducing what varicosities is, right? So, and what are bulbs, right? So since that slide itself is not present, I will just introduce it like that. So the synaptic ends, right? Over here, you can see that there are axon terminals, right? So these axon terminals might be bulb shaped or they might be like a string of, bulb like structures which are varicosities, right? So they might either be bulb shaped themselves, which are known as synaptic end bulbs, or they may be present like varicosities, which is like this, right? So this bulge and then a connection, another bulge, and connection, another bulge, like that. This is known as varicosities, right? So in both these cases, you can see that the synaptic bulb as well as these varicosities, both of them contain many tiny membrane enclosed sacs, which are known as synaptic vesicles, which store chemical which is known as neurotransmitter, right? Uh, so what are these neurotransmitters? You can see that they are different kinds of chemical compounds, right? What do these chemical compounds do? These are basically molecules which are released from a synaptic vesicle, which excites or inhibits another neuron, muscle fiber, or gland cell, right? So whatever effector is present, based on what is the neurotransmitter and based on what is the effector cell, it will either excite that cell, that is, if it is functioning, it will function even more. If it is not functioning, it will start to function, or it will inhibit the function of the effector, right? So if, for instance, uh, I uh, I was already lifting something, right? And there was some signal that, made me think that I have to stop lifting this. So it might have been a inhibitory, signal to the muscles that were lifting it to ask them to keep it back, right? So it will depend upon what is the kind of communication a person is trying to do, right? Many neurons contain two or even three types of neurotransmitters, each with different effects on the postsynaptic cell, right? So it is not important that one kind of neuron will have one kind of neurotransmitter and that will be released, and the postsynaptic cell will respond to that, right? That is not compulsory, right? So just like, for instance, um, there are these multi-cuisine restaurants, right? They will not be limited to one kind of diet, right? Depending upon what is the kind of food the visitor wants, they can give a continental food, they can give Italian food, they can give South Indian food, they can give North Indian, Mughlai, any kind of food they can give, right? So the neurons are like that, right? They have multiple neurotransmitters. They will only secrete whatever kind of information they want the other person to listen, right? So for instance, uh, my phone will have a lot of pictures, right? But I will only upload, some picture to the Instagram or Facebook or whatever social media, right? So that will be, received by whoever is following me, right? I will, if I am sending to somebody like my mother or, brother, then I will maybe send all the pictures from that trip, right? So I have everything, but based on what is the response I want to get or who is the person that I'm speaking to, I will act accordingly. I will use only whatever pool of pictures is required for that particular instance. Then this is another representation for a synapse. I would rather have kept this bracket like this, right? So a synapse in my uh, head would have looked like this, right? So this is actually a synapse. uh, it is a representation which is also showing that there are presynaptic neurons, and in this presynaptic axon terminal, there are synaptic vesicles along with mitochondria. We know that axons have mitochondria, right? So there are mitochondria that are present. There are neurotransmitters within this synaptic terminal that are present. And once these synaptic vesicles that are present, and when these synaptic vesicles they fuse with the plasma membrane, they basically release these neurotransmitters, right? These neurotransmitters are then released into the synaptic cleft, and these synaptic clefts are basically spaces or the empty space between the presynaptic and the postsynaptic neuron, right? Over here also the effector cell is a neuron because it says postsynaptic dendrite. So this is the dendrite and it has multiple kinds of receptors, right? This is a kind of receptor that will recognize this neurotransmitter maybe. Right, and and it is listed as an ion channel. So it might be possible that this particular neurotransmitter is an ion, right? And it is being recognized by this particular receptor, which is why we had studied earlier that dendrites have a pool of receptors that are present on their surface, right? Because there are multiple kinds of effector molecules that exist or neurotransmitters that exist, and they should be capable of recognizing all of them if they want to respond to that particular molecule being released, right? So these are examples of neurotransmitters, right? Some of them you might have heard very commonly, such as adrenaline and noradrenaline, right? So adrenaline is that fight or flight hormone, right? It is produced in stressful situations or when you are very, excited or you are very, not excited, you are very on the edge for something, right? It will prepare your body for that. Noradrenaline will basically be released in a reverse of this situation, right? When the body is basically trying to concentrate, right? Then there are other hormones. Some of their functions have been written over here. It is not only these functions that these hormones perform, right? Even GABA, glutamate, they are even inhibitory in nature, right? So some of these are excitatory neurotransmitters, some of them are inhibitory neurotransmitters, and that sometimes also depends on which cell they are acting on, right? Sometimes on some kind of neurons, if they will be the kind of receptors that they will have will make them excitatory, and in other kinds of neurons the kind of receptors they will have will make them inhibitory, right? So after this, we can look at the structural classification of neurons. We looked at neuron, we looked at the various parts of the neuron, the major parts of the dendrite, cell body, and the axon. We also looked at how, like, what are the parts that are present in dendrites, what are the key features of a cell body, what are the key features of an axon, right? And we also looked at synapse. Now we can look at classification of neurons. So there are two major classifications based on structure or based on function, right? First, let us go through the structural classification of neurons, right? So how is this structural classification done? It is basically done according to the number of processes that are extending from the cell body, right? So, for instance, if there are two processes that are extended, it will be bipolar. If there is one process that is extending, it will be unipolar. If there is multiple processes that are extending, it will be multipolar, right? So the most common type of neurons are multipolar in nature, right? You can see over here. This is the cell body. There are multiple kinds of extensions over here and an additional extension over here, right? Bipolar neuron are most of the multipolar neurons are most of the neurons that are present in the brain and spinal cord, and most common motor neurons are also these only, right? Bipolar neurons are actually generally present, uh, as um, what do we call it? In the retina, for instance, the photoreceptors are kind of bipolar neurons, right? The olfactory receptors are kind of bipolar neurons. They are also present in some other instances, such as the intermediate kind of neurons are often bipolar neurons, right? We will study what they are also. So in bipolar neuron, what happens is there is a cell body. From the cell body by itself, there are two extensions only, right? So that is why bi. So two extension, one is a thick extension for a dendrite and the other is the extension for an axon. Now this dendrite extension then further goes forward and then it subdivides into other kinds of dendrites, right? Multiple dendrites are not arising from the cell body itself, but it is going forward and then subdividing, right? From the cell body per se, there are two extensions only. Then in the unipolar, what happens? These are the sensory receptors are unipolar generally, right? And it is what is happening over here from the cell body. There is one single extension, and then it is further dividing into dendrites and axons, right? So from the cell body, there is one extension only. It is believed that these unipolar neurons, by during development, they are also bipolar, right? And when development is taking place during that process, what happens? That uh, l- this particular extension for dendrite and the initial extension for axon, what happens is that they get fused together, and they then form this unipolar neuron. So as I said, most of our sensory receptors, touch receptors, etc., are unipolar in nature, right? Also, in addition to structural classification scheme that we just talked about, some neurons are named for the histologist who first described them for a particular aspect of their shape or appearance, right? So these you can look at are very distinct looking neurons, right? So because they are so distinct looking, they were described actually by these particular people, right? So Purkinje described this particular kind of neuron, which are present in the cerebellum, and this is what they look like, right? And these pyramidal neurons were named because their cell body is kind of pyramid shaped, and they are present in the cerebral cortex of the brain, right? Okay. Now that we are done with structural classification of the neuron, there is also a functional classification that exists, right? So what will be the functional classification? It will be classified according to the direction in which the nerve impulse or the action potential is conveyed with respect to the central nervous system, right? So there are three kinds of functional classification. The first is the sensory or afferent neurons, right? So what does it mean? They either contain sensory receptors at their distal ends, which is the dendrites, or they are located just after the sensory receptors, that are separate cells, right? Once an appropriate stimulus activates a sensory receptor, the sensory neuron form an action potential in the axon, and the action potential is conveyed to CNS through the cranial or spinal nerves. Most sensory neurons are unipolar in structure, right? So that is what they are doing. They are receiving the signal, generating an action potential so that that signal can be propagated to the CNS. So that is why they are known as sensory or afferent neurons. Now, what is motor or efferent? It is the direct opposite of the sensory and afferent, right? So in this, they are basically conveying action potentials away from the CNS to the effector, right? Be it the muscle or the glands in the peripheral nervous system through cranial or spinal nerves, and these are generally multipolar in structure, right? Then there are interneurons or association neurons. So how do these sensory neurons and motor neurons communicate? It is generally through these association neurons or through these interneurons, right? So these are basically mainly located within the central nervous system between the sensory and the motor neurons. The interneurons integrate or they process the incoming information, sensory information from sensory neurons, and then they elicit a motor response. They will find a response by activating the appropriate motor neurons. Most interneurons are multipolar in structure, right? So instead of bipolar, what they have said is most of them are actually multipolar in structure also. So I have anyway given you example of bipolar neurons. It is present in the photoreceptors of the eye, right? The olfactory receptors also, I think. So this is a representation of all of them. For instance, there was a cactus and somebody touched this cactus, right? So that was a sensory input. It was taken by the sensory neuron. Over here, you can see that it is looking like a unipolar neuron, right? Then it was further communicated to the effector, which is a motor neuron, through an association neuron, which is the interneuron, right? Then this association neuron carried this information forward to the effector neuron or the motor neuron, right, which will further take this signal that the person should remove their hand from there because it is cactus and it will hurt you, or it has already hurt you, right, so that it doesn't hurt you any further. Then comes the neuroglia or the glial cells. Right, so we have talked about neurons also. Now we can talk about the neuroglia or the glial cells. So for today, I think this can be our last topic, right? I can finish glial cells today and then we can just end this session. And we can pick up with the gross brain structure from next session onwards. Anyway, the attendance anyway is low. Yeah, I'll upload this video. Also, we can pick up from the gross structural organization of the brain from the next uh, lesson onwards, right? So, in case of neuroglia or glia, they make up about half the volume of the central nervous system. We told, right, that around 25 times more than neurons are the glial cells, right? Uh, Also, why are they known as glia or glial cells? Because the literal meaning of glia means glue, right? So earlier, when these cells were actually discovered for the first time, it was, and the neurons were already known about, right? So it was thought that these are the cells that are holding the neurons together, right? Or they are acting like glue so that they're holding the neurons in position. So that is why they were named as glia, right? But over time they themselves have been studied extensively and it has been seen that multiple of these glia actually have very specialized functions, right? They are not just glue cells or supporting cells, right? They have very specific functions of their own. So those are the functions that we will discuss today. So generally, the neuroglia are smaller than neurons and they are 5 to 25 percent times more numerous, right? Although they are smaller, they are much more abundant than the neurons. So in contrast to neurons, the glia do not generate or propagate action potential, right? That is only a function of the neuron. And these glia can also multiply and divide in mature nervous system, right? Unlike the neurons, they can actually multiply and divide, even in the adult nervous system. There are six types of neuroglia, four of which are present in the CNS only, and the remaining two are present in the PNS, right? So in the PNS, there are satellite cells and Schwann cells, and in the CNS, there are these four types, which are microglia, astrocytes, ependymal cells, and oligodendrocytes, right? Sorry. So this is a representation of the neuroglia in the CNS, right? Over here, these are like the meninges. Then you can see the oligodendrocytes, the microglia cells. Neurons are not the glial cells, but you can see that these glial cells are basically smaller looking than the neurons and they are surrounding them, right? Then there are astrocytes that are present. Multiple kinds of astrocytes are listed over here, fibrous and protoplasmic, which we will not go into the detail of. Then ependymal cells you can see over here at the bottom, right? Oligodendrocytes has been marked, right? So all these various kinds of glia are, see, they are taking up so much space over here, right? Along with the neurons, so many of them are also present, right? Okay, so and this is the representation of the glial cells present in the peripheral nervous system, which were the Schwann cells, over here, right, in unmyelinated axon. This is where the Schwann cell is. In myelinated axon, this is where the Schwann cell is. And the Schwann cell in myelinated axon and satellite cells, right? So the first kind of glial cell that we can talk about is astrocytes. Now these are one of the most important glial cells actually. Why? Because they have so many vast range of functions, right? So these are basically star-shaped cells like this. They may have processes and are the largest and most numerous of the neuroglia. So they are larger in size and they are also the most numerous of the neuroglia, and they have a diverse role in the brain, right? The processes of astrocyte make up contacts with the blood capillaries, neurons, and biomatter, a thin membrane which is found in the brain and the spinal cord. Now, what are the different functions of this astrocyte, right? Some of them have been listed over here, that if asked in a healthy condition, what does the astrocyte do? It basically is involved in synaptogenesis and synapse maturation. It is also involved in modulation of synaptic transmission. It is involved in trophic or metabolic support, right? Why this? Because it is also directly, uh, attached to the blood vessels in the brain, right? In fact, astrocytes is one of the components that form the blood-brain barrier, right? So it is forming a barrier kind on the blood vessel, right? So because it is forming a barrier on the blood vessel, it is being very extremely selective about what is going into the blood vessel of the brain and what is coming out of the blood vessel of the brain. That is why that blood-brain barrier is present, right? So because it is so curated, that is why it is responsible for the trophic or metabolic support, because this is coming via the blood generally, right? Then it is also important for uptake and recycling of neurotransmitters. It is involved in that as well. It is responsible for maintenance of blood-brain barrier integrity and regulation of blood flow. As I told you, it is a part of the blood-brain barrier itself. Then it is responsible for water and iron homeostasis again, because it is directly also in contact with blood and also in contact with neurons. It has multiple transporters that are present on its end feet over here, right? So they will help maintain water and iron homeostasis. Also, it has a neuroinflammatory kind of role. That is why it is involved in phagocytosis and synapse pruning. Right? So, phagocytosis, as in, even if there is debris, they can maybe remove it. Right? And example of phagocytosis is also given during synapse pruning. So, synapse pruning is actually something that happens when we are developing, right? So at the time of our development, there are multiple neural connections that are there, right? Not all of them are required. So a synapse pruning goes on, because if all of them are kept, then we will not be able to accommodate any new information or memory or skill as we grow up, right? So we need only the connections that are necessary when we start off. So that kind of synapse pruning is done with the help of these kinds of phagocytic, machinery, one of which is also astrocytes, right? Now, because I have picked it up from a review article which was titled An Overview of Astrocyte Responses in Genetically Induced Alzheimer's Disease Mouse Models, you can see that in case of a neurodegenerative condition such as Alzheimer's disease, what happens to an astrocyte, right? So there are some changes due to which it becomes reactive. When it becomes reactive, you can see the morphology changes a little. There are much more branches over here now. Then it also, starts the phagocytosis and degradation of amyloid, right? Because it is actively involved in that process, the interaction with the vessels is going down, right? So if the interaction with the vessels is going down, then what happens? The blood-brain integrity of the blood-brain barrier integrity is actually lost. Then it also might cause neurotoxicity because of loss of trophic support. Then the again the neurotransmitters recycling, that capability is going down, due to which what happens? Excitotoxicity. Now, what is excitotoxicity? That some neurotransmitter was present, right? Now, after a neurotransmitter has been released, within a few seconds or minutes, what happens that it needs to be removed from that place. Otherwise, the effector cell will always keep responding to it if it is not removed, right? So removal process is done. Either some neurotransmitters are taken back in the presynaptic neuron through certain receptors, or some cells like phagocytotic cells or some enzymes act upon them to break them down, right? So one of them is the astrocyte also, as we studied, right? So if the astrocyte is not functioning to its capability, then the neurotransmitter, it will not degrade, right? That neurotransmitter will remain there and the effector cell will keep functioning. So over functioning will also create a type of toxic condition that is known as excitotoxicity, right? Then it will also lead to cytokine secretion, which will lead to more inflammation in the brain. It will lead to altered calcium transients and cap channel communication. So calcium is actually responsible for a lot of signaling in the brain. And if that is altered, signaling will be altered, right? Also, it will change to lead to morphological changes, due to which there is increased GFAP. So GFAP is a protein which goes up in the astrocytes that are actually reactive, right? Glial fibrillary acidic protein, right? So this is a marker for for astrocytes actually, and loss of aquaporin 4 polarization, meaning that these aquaporin 4 are basically present on the end feet or the feet of the astrocytes that basically lie on the blood vessel, or they are helpful in forming the blood brain barrier, right? So if the astrocyte is reactive, there is loss of polarization or proper arrangement of this aquaporin 4 channels, that is the end feet of the astrocytes on this blood vessel, right? So these changes actually occur. Then there is oligodendrocytes, right? So oligodendrocytes resemble astrocytes but are smaller and contain fewer processes, right? So these are oligodendrocytes. What is the main function of oligodendrocytes? They are responsible for forming and maintaining the myelin sheath around CNS axons, right? So in PNS, somebody else is taking care of this. In CNS, what part or which cell is forming this myelin sheath? It is basically the oligodendrocytes, right? Yeah. Then we can look at the microglia, which is yet another kind of glial cell, right? These neuroglia are small cells with slender processes that give off numerous spine-like projections, right? So over here, this is still a cartoon kind of a view of microglia. If you actually look at a microglia, you will see that there are very sharp and abundant processes of microglia that are present if it is in a normal state, right? Because these microglia cells basically function as phagocytes. So these are the resident macrophages in the brain, right? So as I was saying, Kupffer cells are resident macrophages in the liver. Similarly, Dutch cells are resident macrophages in the eye. There are resident macrophages in the brain also, which are the microglia, right? So macrophage, because it is going to surveillance for carry forward a surveillance all the time for any debris or any kind of pathogen or any kind of harmful substance in the brain. So that is why it will have these numerous processes, right? So that is what the microglia looks like normally. But in cases where there is activation of a microglia, or there is a chronic inflammatory condition that exists, what happens is the cell body of the microglia grows and it loses its cell body gets swollen and it loses a lot of its projections, right? Generally there is a case where there is hyperamification also, but mostly it becomes a cell body becomes very swollen and it loses a lot of processes, due to which what happens is that the surveillance is not being able to be done nicely, right? So they are not really acting as good resident good macrophages, right? Then we come to ependymal cells, which is the final kind of glial cells that is present in the central nervous system. So, what are these ependymal cells? As you can see over here, they also have a very stark structure, right? So, these are cuboidal to columnar cells. Generally, they are either cuboidal or columnar, and they are arranged in single layer that possesses microvilli and cilia. Right, so it contains these kind of hair-like structures, which are the microvilli or the cilia. These cells line the ventricles of the brain and canal of the spinal cord. Right, so ventricle is basically the hollow space that is present in the brain, and central canal of the spinal cord is also the hollow structure in the spinal cord, and these hollow spaces and hollow structure is filled with cerebrospinal fluid. Right, so these spaces are filled with cerebrospinal fluid, which protect and nourish the brain and the spinal cord. Right, functionally they produce and possibly monitor and assist in circulation of cerebrospinal fluid. So they are producing cerebrospinal fluid, and they are also helping it circulate because of the presence of villi and the cilia that are present. They also are responsible for forming the blood cerebrospinal fluid barrier, right? That is what they are doing. Then there were two kinds of glial cells that were present in the peripheral nervous system, actually, right? They were the Schwann cell and the satellite cells. So let us go through them also quickly so that we can wrap up the session after that. So what are the Schwann cells? These are the cells encircle the PNS axons. So they are similar to the oligodendrocytes then, right? Like oligodendrocytes were also creating this myelin sheath, right? Similarly, Schwann cells are also creating the myelin sheath, but they are creating the sheath in the PNS axons, not the CNS axons. Like oligodendrocytes, they form the myelin sheath around the axons, right? A single oligodendrocyte myelinates several axons, but Schwann cell myelinates a single axon. That means that in the CNS, one oligodendrocyte could myelinate multiple axons, right? If we go back to that representation, I can show it to you also, right? So there was this one oligodendrocytes and multiple cells it could myelinate, right? Over here, however, they are saying that one Schwann cell can myelinate one axon, right? So that is what it will do. Schwann cells participate in axon regeneration. We talked about this, that axon regeneration is actually possible or more likely possible in PNS than CNS, right? Which is more easily accomplished in the PNS than the CNS. Then we come to the final one of the glial cells, which is the satellite cells. So these satellite cells, which you can see over here, are flat cells which surround the cell bodies of the neurons of the peripheral nervous system, peripheral nervous system ganglia, right? So, like there are cell bodies or nuclei in the collection of cell bodies or nuclei that are present. Okay, I think there was a call drop. I will just share my screen and we can just finish this. Yeah. So we were talking about the satellite cells actually, and how these are the flat cells that surround the cell bodies of the neurons of the PNS ganglia, which is corresponding to the nuclei or the cell body in the CNS, right? Beside providing structural support, these satellite cells regulate exchanges of materials between neuronal cell bodies, which they are surrounding basically, and the interstitial fluid, right? So that is their major function. So with that, we can just end this session, right? Anyway, we are facing some issues with the network now. So we can stop the recording, I think, and we can save this session.